Explore the history, relationships, expertise, and data that go into ensuring Stine growers get maximum yield potential. This is the Stine Seedcast. Here's your host, David Thompson.
Hello, and welcome to the Stine Seedcast. I'm your host, David Thompson, National Marketing and Sales Director for Stine Seed Company. We've got another great episode lined up with special guests, expert insights, and discussion on everything you need to know about maximizing yield potential. We have two special guests for today's episode, Sekar and Bruce Held. Sekar and Bruce are research scientists at Stine Biotechnology at our lab up in Ames, Iowa. Welcome, Sekar and Bruce.
Thank you, David.
Yeah, hello, David.
Glad to be here.
So today we're going to talk to them about the work that goes on at Stine Biotechnology, get a little bit of background about the lab, and talk about some of the developments they've had to help our grower customers maximize yield potential and have flexibility in their operations. We're going to learn how they use advanced proprietary technology to deliver the seed industry's best corn and soybean germplasm. So let's get started. Gentlemen, just to get us started, I thought we'd provide a little bit of background for our listeners, biographical history. So maybe Sekar, to tell us a little bit about your background.
Okay. Thank you, David. I grew up in South India, and I came to the U.S. quite a long time ago to do a PhD, and I went to New Mexico State University in Las Cruces, New Mexico, got my PhD in biochemistry, and I did a little bit of postdoc in different places and worked with some ag bio industries in the U.S. and stayed here for so many years. And then returned back to India, South India again, to a major university there and taught biotechnology for about 10 years. And I became a professor and head of a department there. And I had 12 PhD students completed PhD under my guidance. And then I returned back in 2002 to United States, back again to Stine Biotechnology, and that's my background.
Oh, perfect. How about you, Bruce?
I was raised on a farm in west central North Dakota. It was in Morton County, actually. And there's something kind of interesting over the last 35 years that's happened in Morton County. When we grew up, there were no soybeans planted in Morton County. And now there are. And in fact, I think Stine has an independent sales rep that operates out of Morton County. So I find that interesting over the years how the soybean acres have changed. And so after that, I went to college at NDSU, North Dakota State University. And it was there, I took a genetics course and I learned for the first time about this process called transformation and how you could transfer a gene from one species to another. And and that was the first transgenic plants were produced in like 82 and 83. So it was just new stuff, and it caught my attention. And I thought, I'd like to do some research in that area. So I continued to do some research. I got a master's degree there, and I came to Iowa State and got a PhD. And then from there, I did a postdoc at ICI Seeds and then landed at Stine Biotech.
Okay, awesome. Thank you. So I guess that's a good place for us to start. I know, I came to Stine in 1998, and Stine Biotech already existed, right? So I know, Bruce, you were there, and I'll kind of open it to both of you, but certainly, can you give us a little bit of background about, kind of the formation of Stine Biotech and how that all came into being?
Yeah. Stine Biotech started in 1995, and Martin Wilson was the first director, and he was instrumental in getting that started along with Harry. But there's a little background to that. Harry had already been funding some soybean biotech research at what was then ICI Seeds, and that's where Martin was, and he was directing that research. And in fact, the type of work they were doing, they were creating genetic variation using tissue culture, and so they wound up with a product, actually. It was a variant of 2250, and I believe the line was called 1580. But it was very much like 2250, as I understand, but it was an earlier maturing variety, and so they could grow it up further north. So that was the first product out of that work. And then, later on, as events turned out, Martin was let go, from ICI Seeds. And so he approached Harry, and I think Harry, was keen in starting a lab. And it was 1995 then, and it was the year before the commercial release of Roundup Ready soybeans, so I think the timing was right for it,
And most of us in ag hadn't heard a lot about biotech seed at that point because, like you said, the shot heard round the industry had not arrived yet, right, for Roundup Ready.
That's exactly right. And so, Martin approached me, and he, I had a temporary position really, and he was like, "You want to help start the lab, And so there was about 5 people who were in that startup group, if you will. It was Martin, it was Liming Ho, and Janelle Eby, and there was Carol Lunau. Yeah, so there was 5 of us, and I remember it being an exciting time, but I also remember it being a daunting time, because we didn't have any genes to work with. We had to get some equipment and start from scratch, And we wound up at the ISU Research Park in Ames, which has been good. We've been there now going on 28 years, but we have a good relationship, with Iowa State. They offer some services for us, that we can utilize, DNA sequencing and other services and library, and we can interact with some of the people there at Iowa State. So the location's been good, and that's where we started, and that's where we still are.
So what I'm hearing you say, your path to Stine kind of came through Martin, your relationship with Martin.
Exactly.
And I guess what was the motivation for you to make that jump at that time?
Oh, I just looked at it as an opportunity. I guess there was a lot of risk and that kind of thing involved, but I think I was young, and it just looked like a good opportunity. And also, it's kind of interesting, as a graduate student, I wound up working on some soybean plots, and quite frankly, I'd never heard of Stine, okay? And I was working on these soybean plots, where I think they took a 15-foot bar and you had to lay them beside the rows, and you had to weed them back, and they're going to do yield trials at Iowa State, And I remember asking the professor in charge, I said, well, who do you think's going to, win these trials? And he goes, I think Stine's going to. And that's the very first time, I'd heard of Stine, so I wasn't that familiar, actually with it. But that I had that in my back of my mind yet, about that experience as a grad student.
Okay.
Yeah.
So, Sekar, you came to Stine Biotech in 2006, so just 2002? Okay. And so I guess what was your journey to Stine Biotech? Yeah. What led you there?
Okay. I was in India, as I was mentioning before that. I worked here and then went back to India. I was a professor there. For whatever family reasons, we thought we will return back to the U.S. But I've heard about Stine quite a long time. Prior to that, I was working with a biotech company at Madison, Wisconsin. Harry, at that time, had a lab in Madison, and it was called Stine Microbials. And one of my friends worked there as a director, and I had close contacts with him for a long time while I was in India too. And so when I wanted to return back to the U.S. again, I thought of Stine as a possible choice and approached Harry, and I got an offer to work with him, and I've been here since then, 2002. And by 2006, Martin was gone from the Stine Biotech group, and I became the director of the organization. I've been holding that position ever since.
Awesome.
And Bruce mentioned there were 5 people. Originally Bruce, Martin, Carol, Janelle, and Liming Ho. 2 are still here, Carol and Janelle. And then we are now, again, me, Sekar, and Sume and Kyle. We are the 5 people, and we've been that for a long time now as a group.
Well, and I know you do a lot of fantastic work up there, and a lot of material goes through there. So let's talk a little bit about the mission up there. I mean, what is the purpose? What is your lab doing up there as it relates to Stine and our product line?
Yes, the main motto of Harry and Stine is Stine has yield, And anything to do to improve the yield of corn and soybean through genetic manipulations is what we do. And basically, we look for genes that we can introduce, traits that we can introduce into corn or soybean through tissue culture, through genetic manipulation. If you can introduce and show such introduced traits, improve the quality of the crop, and finally gives a better yield, and that's what our main goal is. And so we do a lot of we search for novel genes associated with novel traits. And primarily herbicide resistance is one of the major, just like everybody else has.
Sure.
But we work with various additional traits that eventually can give better yield potential to the crops.
And is the work at the lab focused primarily on corn and soybeans?
Yes, primarily on corn and soybean, but we also have a smaller project. We work with a plant called Monarda. That's an oil. It's a natural oil producing plant, essential oil producing plant, and Harry works with that, and we improve the quality. Monarda is it's a wild plant variety that grows in Iowa and several other Midwest and even Eastern states. And Harry has been working on it for a long time to improve the quality of the plant for improved oil content and a certain oil profile. He's been looking for varieties of Monarda that has certain oil profiles. And through just continuous culturing and selection, he has been looking for varieties that has the oil profile that he is eventually interested in.
And Monarda may someday get its own podcast because such an interesting crop and an interesting plant. So I'm glad you brought that up. We may want to circle back around to that. So over the years, and like I said, I know you've done a lot of work there at the lab. What are some of the things that you would list as accomplishments you guys have had during your time there?
Well, like I mentioned, when we started, we didn't have any way of putting, say, the genes into a plant cell. And at the time, there were primarily two ways of doing that, and that was Agrobacterium and the gene gun. So we needed, and those methods were wrapped up in patents, usually by big multinational companies, and so we needed a way to introduce genes into cells so we could have freedom to operate.
And Bruce, while we're on that topic, for the listeners, I think it might be good if we give a brief description of what those transformation methods are, right? For maybe the uninitiated, Agrobacterium would be.
It's a biological means of transferring DNA into the plant cell using Agrobacterium itself.
Right, so it's a bacteria that basically uses.
Agrobacterium is a plant pathogenic bacterium.
Yep.
It has a way of, when it infects, usually dicot plants, when it infects, it does infect through wounds in the plant. And it gets through the plant tissue, through the wounds in the plant. And it has a way of sending it a small piece of its own DNA into the plant, and makes the plant to proliferate at the site of infection, causes a gall in the tissue, where it enters in, it produces a gall. It's called a crown gall bacterium, that causes, several dicot plants. And much later, this has been known for a long time, but by early 1970s or so, people found out the mechanism by which it manipulates the plant cells to grow like a gall. And it's by the way of introducing a small piece of its own DNA into the plant. And asks the plant or makes the plant to make the compounds that it wants, it can utilize and grow on. So once that mechanism was found out, then people thought, okay, if we took out whatever the toxic components that are present in this small piece of DNA that goes from the bacterium into the plant to make the plants to grow as a cancerous tissue, if you knew the mechanism and figured out which part of its DNA causes all this proliferation, once they knew that, they removed all that. Instead, you can introduce whatever DNA you want into that little piece of DNA. that goes from Agrobacterium into the plant. And like a Roundup resistance gene, like whatever trait that you want to introduce, if you introduced into that little small piece of DNA that goes from Agrobacterium into plant, then you put that DNA into Agrobacterium will deliver into the plant. And that's how Agrobacterium-mediated transformation works.
So that way you're using this bacteria's own natural mechanism. You're basically hijacking it to deliver the DNA that you want to deliver into the plant.
Natural plant genetic engineer is Agrobacterium. And that's what we use.
And several of the crops probably that we're used to today, right, were probably work was done through Agrobacterium. Is that correct?
Yes, and in fact, Agrobacterium was the first means used to transform plants. And so, like Sekar mentioned, it was mainly a dicot pathogen. And so when, people wanted to transform corn, at that point in time in history, Agrobacterium wasn't working for corn. So that led to the gene gun.
Okay.
And the gene gun is really a mechanical way to introduce what we call naked DNA. You use particles, usually around 1 micron in size or a little smaller. You coat them with the DNA and you literally blast those particles into the plant cells. And that was, used in a lot of monocots, especially at first, but it was used in some dicots too. So those were the main two methods. There's some other methods that are around, like they called it whiskers and electroporation, but I think, those were the main two at that point in time.
But as you said, those were the two main ones, but due to different patents and things, a lot of companies had fairly controlling interest in those projects. So how did you guys respond to that?
Well, we thought we needed a novel way of doing this, getting the DNA through the plant cell wall. And so we knew of a professor at the University of Chicago, Dr. Loren Metz, and he had this idea of using an aerosol beam injector, and he had the idea of accelerating this without particles. It's a law of physics where if you generate a mist, in an upper chamber and then draw it through a small nozzle into a vacuum chamber, and at that point in the nozzle, these droplets would be accelerated, they said, to supersonic speeds. And so it was a different mechanism of acceleration. Didn't utilize any particles, and it looked like it would work. It might have a chance of working. So we worked with him in the early days, and we made some improvements to it, and I think we reduced it to practice, as they say. And so we filed some patents on that, and from that time on, we had freedom to operate, with that device. I should also mention we worked with an engineer at Iowa State too. His name was John Jackman, and he really helped us in prototyping and introducing some electronics. We had a stage that was controlled with a computer and with electronics, and he made it a really nice, workable, usable system for us.
So aerosol beam injector really went from theory to
to practice.
In your lab.
Yeah.
And you use that method. I assume you probably use the other methods as well at various times, but depending on what the right approach is.
Exactly.
Okay.
Yeah. Mm-hmm.
So the aerosol beam injector, what does that bring to Stine Biotechnology? What are the benefits of having that in your arsenal?
Well, I think the main benefit was the freedom to operate. And with all these methods, some of them, there's disadvantages and advantages.
Sure.
I think, for instance, Agrobacterium, one of the main advantages was it had what we called a reduced copy number of integrations, in the genome. It was a benefit of Agrobacterium. But nowadays, with gene editing coming on and things like that, I think naked DNA delivery methods have become a little bit more in favor, because you can introduce maybe RNA, just RNA. You can introduce protein, perhaps, and it's actually beneficial to do that. And you can't do that with Agrobacterium, just introduce RNA alone or a protein alone. So an aerosol beam injector would have the advantages of any naked DNA delivery system. I think to deliver RNA or oligos or that kind of thing, it would be good at.
And when you talk about freedom to operate, I know one of those things is, like you said, with if you're using one of those other methods, you're kind of having to play in the other company's sandbox, so to speak, right?
Oh, yeah.
And so they may say to you, well, we like your project, we think that's okay, or they might say, well, I don't know if I don't know if we want you doing that. So having the flexibility to use your own method,
Oh, yeah.
and even the ability to say, well, if you don't, we have that flexibility, right? And that's really kind of the advantage that you have with that.
Yeah.
With that tool.
Yeah, and in the early days, we attracted some gene discovery companies. You know, they were in it. They had some genes that they wanted to test. You know, we could say to them, look, we can introduce them for you, and we can test them, and there's even a pathway to market for you. You know, if they really work. You know, so you know, it gave us that opportunity too to, you know, work with others.
Because would it be fair to say that some of the companies who are in what I'd call gene discovery, that's all they do, right? I mean,
Yeah.
they don't have germplasm. They don't
Right,
have
exactly.
transformation. They don't have
They don't have marketing.
a marketing arm to even if they make a product. So
Right.
in that sense, Stine Biotech kind of provides a one-stop shop of sorts. Okay.
Yeah.
And we talked a little bit about, I think Sekar, you had mentioned you do some gene discovery or you're involved in some gene discovery. So we talked about that just now. I guess I know there's companies that specialize in that. So how do you find the genes of interest that you guys are looking to get involved with?
It's a very complex process, and if you know what you're looking for, like herbicide resistance, let's say, it's a little easier to talk about, or insect resistance for that matter. Insect resistance is fairly well known in nature. There's a bacterium called Bacillus thuringiensis. It's called BT for short. And BT incorporated plants are known for a long time now. You know, the BT bacterium produces a protein, and which is insecticidal in nature. And there's a variety of BT strains available in nature. Some BT proteins kill mosquitoes. Some BT proteins kill beetles. Some kill Lepidopteran, this larva, you know, various forms of larva. So there's a wide variety of BT strains available in nature. So if, for example, we are interested in a corn rootworm killing Bacillus thuringiensis, BT, we needed to screen a variety of, you know, from various sources, identify BT strains, and then test them on corn rootworm larvae. And if you're lucky, you know, this has been done throughout the world with the various major biotech companies working on it. It's very difficult for you to really hopefully get one, but if you did end up getting a Bacillus thuringiensis strain killing a corn rootworm larvae, you go back to the organism, the BT, and figure out what gene from BT kills that corn rootworm larvae. And once you identify that gene from the bacterium, you dissect that gene out from the bacterial genome chromosome or bacterial DNA, and take that piece of DNA that is responsible for making that protein that kills the corn rootworm larvae. Take that, identify that. These are all a lot of basic research you needed to do first. Once you identify that, then you take that gene and then put it into in a DNA, what we call a construct, that is, you know, where you introduce the DNA into another DNA piece of DNA that can be introduced into the plant cells. So you take the gene from a bacterium, adapt it in such a way that it will be expressed, or the protein will be produced from that gene in a plant cell. You make all the tweakings in test tube, in vitro it's called. So you do that, and then once you come up with a DNA construct that has that BT gene that is now ready to be delivered into a plant cell, then pass it on, let's say, to Bruce, and he, through aerosol beam injection, introduces into plant cells. And eventually we come up with plants that has the gene in it, and then we test those plants that come out of the transformation for their ability to kill corn rootworm larvae.
If I may inject something here, actually Sekar is a BT expert, and he's actually widely known for having isolated the first coleopteran active gene from BT, and it was called Cry3.
Yeah. What is Cry3BB? You may be familiar with the
Right.
that the Syngenta has. I'm not sure what I can. So other companies have coleopteran resistant. Those are beetles. So those we were one of the very first ones to identify coleopteran potato, Colorado potato beetle specific. That's toxic to Colorado potato beetle, but that's the first time ever a BT that is killing a coleopteran larvae was identified by us. We also were involved in, for the first time, identifying BT protein that kills mosquitoes. And we do have a patent on, I do have a patent on that, and that's called a Cry4 protein, Cry4 gene or Cry4 protein. Cry3 is the coleopteran one. I was involved in discovering that, and
oh wow,
so
that's very impressive. That's cool.
Yeah.
You mentioned something I want to go back to, talking about constructs, because as someone who's not a scientist, I was kind of very interested in this. My uninitiated view of things were, you find these genes and you kind of open the hood and you throw them in and everything's good. But as you just pointed out, you kind of have to know what it is you're trying to address, right? In the case of corn rootworm, I've heard you say, it does no good if you put that gene in and it expresses in the tassel, for example, because that's not really where you're trying. So talk a little bit about making the constructs. I think that's something that's important for people to understand. You don't just throw it in and it does what you want it to do.
Yeah,
right?
A very good example, as you are saying, this corn rootworm toxic protein that is made by BT. So the gene, there is a gene in a certain type of BT organism, which is called which may be responsible for killing corn rootworm larvae. So we take that gene from the BT bacterium, and then you needed to. The way a gene works is a gene has 3 functional parts to it. The very first beginning of the gene is called a promoter. And then the middle part is actual gene that is responsible for making a protein. And the last part is called a terminator or ending part. It's a termination of the gene. So the promoter and terminator elements are so specific what is in the BT organism for producing the BT coleopteran toxic protein. The promoter in BT will not be recognized as a promoter in a plant. So also the terminator that's present in the original BT genes, coleopteran gene, will not be recognized as a terminator. So if you took the whole thing and put it into, just like that from BT into plants, it will not be making the BT protein that is responsible for killing coleopteran larvae. So you needed to dissect the promoter and the terminator elements out of the original gene and instead introduce a promoter and terminator elements that is specific for a crop that you are interested in. Even within the plant kingdom, corn, a monocot, soybean, a dicot, they do have different specificities as for what they can recognize as a promoter and a terminator. And there may be some promoters and terminators that may work in both crops too. But in addition to that, your question is well-founded because, the corn rootworm feeds on the root, So you do not want to put a promoter that is expressed in a leaf or a tassel if you want to kill the corn rootworm larvae that is feeding in the roots. So you needed to introduce a root-specific promoter if you're taking the coleopteran, the corn rootworm specific protein that you want to express into plants, into corn plants. You need to have a root-specific promoter and a BT gene that is responsible for the corn rootworm larvae, and then a terminator that can really stop the expression of the gene at that point. So those are very specific elements that you have to introduce at the very beginning of the gene and the ending of the gene, as well as you have to modify the gene per se in such a way that the genes, even though without changing the amino acid composition of the protein, you needed to modify what's called the codons. Plants have a different monocots have a different specificity for each codon. Codons are 3 bases in a DNA that encodes for a given amino acid. And so what is present in a bacterium for a specific codon for a specific amino acid, you cannot, may or may not be recognized or recognized well in a plant. If you want to maintain that amino acid in that position, you have a little way of adjusting the codons in the bacterial gene so that you can tweak it in such a way it will be recognized much better in a monocot or a dicot. So you needed to do a lot of modifications at the beginning of the gene, actual gene, at the end of the gene. And once you make all the tweakings, then it becomes a complete cassette that is ready for expression in a plant.
Mm-hmm. So so there's all these different components that go into it. You have to find the gene, then you have to kind of, the gene sequence you want, then you have to modify it slightly to fit the organism you're going to put it into.
Exactly.
But you also have to have the right, like you said, promoter and the right, terminator on the end of it. If you don't do any of those parts, if you screw up one of those parts, the result is probably not going to be what you want it to be, right?
Exactly.
So, and like I said, I think that's kind of a misconception sometimes among the uninitiated, is that it's a lot simpler than that. Of course, we all know that, at the base level, they're all, all the sequence, all the genes are similar in their composition, but how they're arranged is very unique, right? So while we're talking about things, novel introductions there at Stine, I know there was some work around an event called HCEM 485. I know, Bruce, you had some involvement in that. I wonder if you could kind of elaborate on that for the listeners.
Yeah, that event is what we call a cisgenic event. And so you have transgenic events. That's where you take a gene outside the species you're working on and you put it in. But with cisgenic, you're dealing with a gene that comes from the same species that you're working with. So in this case, we took a corn gene and put it back into corn. But this gene, we mutated. We performed targeted mutations in 2 different locations, and those mutations gave it resistance to glyphosate. And we went to the corn genome, we found the EPSP gene, and we used all of its regulatory elements that were in the corn genome as they were. We used the promoter, we used the terminus, and we also used the introns that came with that gene. These are intervening sequences. So the complete genomic fragment we used, and we introduced it with the aerosol beam injector alone, with nothing else, no bacterial sequences or anything. And we got glyphosate resistance corn, and it was cisgenic. And I remember, at the time, this really fell outside of the regulatory, scope. And in fact, we went and visited them, the U.S. regulatory authorities, and we said, here's what we have. It doesn't fit into your regulations, really, would you recommend for us, what to do?
Because at that time, what you're describing is corn genes in a corn plant.
Yeah.
The only, if you call it alteration, is this mutation, which, if I'm correct, is a known mutation that to some extent can occur naturally.
Yes, exactly.
Right? So virtually indistinguishable from conventional corn. So you take it to the regulatory agency, say, this is corn.
Yeah.
What do you want to do with it, right?
Yeah, exactly. And they listened to us, and they were like, you're right. This actually really technically falls outside of our regulations. We don't know quite how to handle this, but we would recommend that you put together a package, kind of a minimal package, and send it through our regulations, they recommended that. So that's what we did. We went and put together a kind of a minimal regulatory package, describing the event and doing some of the tests that they wanted us to do. And so actually, it went through the system and it was, approved. And we also went through the Canadian system while we were at it. So it was a unique event. It was kind of fun to work on.
Yeah.
Yeah.
And, currently we don't commercialize it. It's obviously you'd have to have all the other global regulatory approvals that come with it. But I thought the part I thought was interesting about that story is, like you said, it really didn't fit the conventional, story of what the regulatory rules were around. I mean, here you have glyphosate corn,
Yeah.
essentially occurring naturally, right? And I thought that was really, really novel. And it forced a conversation, I think, within our industry, at least at that time, that needed to be had.
Yeah, and also by having that selectable marker available to us, we went and filed a patent on it, and we were able to then use that as a platform to combine other corn genes to it, and so make more cisgenic plants, because it works so well as a selectable marker. So if you wanted to, find another trait, say maybe it was for, let's say yield increase, although that's, a tough trait to handle with one gene, you could connect it to there and call it a cisgenic event again. Yeah.
Yeah, that's really interesting. Talk to me a little bit about, the work that's going on right now, the work that you're doing today at Stine Biotech. What are some of the things that are, taking your time, the things that you're working on, trying to deliver for, eventually for growers? One of the things you talked about, your dedicated lab of people, you got a great team there, but, as we talk about all the time, you only have so many people and there's so many hours in the day. One of the things I know you guys rely heavily on is collaborations with other companies so that you can play to your strengths and bring in other resources. So can you talk a little bit about some of the collaboration work that you do?
Yeah, we have done quite a bit of work with other companies and came up with products through these collaborations. The first one we did was the product GT27, soybean, and we worked with Bayer at that time, and we were involved quite heavily in looking at the biosafety information that, when you have an event, it was called FG72, the original event was called FG72. And in order to get through regulations, you have to do a lot of work, biosafety and agronomic and yield analysis and all that you needed to do in field and in the lab. And we were involved in reviewing and looking at it carefully and making all kinds of suggestions and coming up with the final event or events that may be taken forward. And in this case, the FG70, before FG72, there was an additional event called FG74. It had some pitfalls, so we ended up finally zooming in on FG72 event, which is GT27. Later, again with Bayer, we made a breeding stack with FG72 and LL55. And we were involved in the regulatory package processing. And the feather on the cap is our collaboration with the Dow and produced E3. And we were involved right from the beginning through the end in event selection. And in order to select an event from all these, whatever trait that you are trying to put into, let's say, soybean, you have to look at a lot of different events, choose the best event to take forward. And in order to do that, that really depends on various results from various analysis. Biosafety, and then agronomic traits, and then yield. And eventually, do all that based on all those results. Bruce and I was instrumental in, along with the scientists of Dow, figuring out the main event for the triple stack in soybean as E3 was our. We were quite heavily involved in selecting the E3 4406 event and then other backup events for that triple stack trait. And so we had been working with, again, a lot of different companies, particularly with Bayer and Dow, and coming up with all these products over the years.
Well, and those are pretty good name drops, Sekar, because for our team, LiberLink GT27 has been a big product, and now, of course, Enlist E3, huge product for our sales team and for the customers that we work with. So to know that you guys had a part in that is really, really kind of fun and exciting. So with that feather in your cap, I guess what kind of things are on the horizon? What are you guys looking at now going forward?
We are working with, right now, one of the major efforts that we're putting in is developing soybean and corn plants through gene editing. And we are working quite a bit on coming up with edited plants rather than GMO plants. I suppose you see the distinction. Instead of introducing a whole gene from a different source or from the same plant as a modified version of it or whatever into a plant, you go and edit the plant's own genome and then come up with possible traits, improve a certain trait that's already present in the plant, in the crop, but through editing, you tweak it, make it better. And you can editing has its own pluses and minuses. The minus could be that you can tweak only so much. You may have to introduce a novel gene in order to really come up with a totally novel trait. But you may tweak a lot of different genes in such a way that you can improve any given trait slightly, or possibly you can do that, But for example, Bt gene has insect resistance property, and that is not present in plants, no matter how much you tweak.
Exactly.
You cannot come up with that.
Yeah.
You cannot come up with that. Or a herbicide resistance gene coming from glyphosate is a totally different story, but novel herbicides, various other, let's say 2,4-D, is not present in plants, or for example, very HPPD, maybe HPPD may be manipulated.
Yeah.
I don't. So anyway, 2,4-D type resistance property cannot be manipulated so easily within a plant, but you can do a lot of other things by tweaking the plant's own genome through gene editing. But the main reason for going through that route is gene-edited plants are normally exempt from regulation within the U.S. And Europe has still not passed a mandate or rule that it's exempt yet. But it seems like it's only a matter of time before Europe goes with that. We don't know that. We have been looking at Europe for the last several years, hoping that they will exempt it, but so far it has not been the case. So we are looking at novel traits to come up with novel traits through gene editing in both corn and soybean.
And I don't want to minimize your work. I may be unfair because I have told groups before, the science is not the hard part. Sometimes it's the politics that's the hard part, right?
Yeah.
So I think what I hear you saying is, that gene editing may provide a more streamlined pathway from a regulatory perspective to be able to bring, because these products these days take 10, 12 years to come to market, and hundreds of millions of dollars to get them into the marketplace, into farmers' hands, and so that's a real struggle. So anything we can do to improve that, I think, is a good thing, right?
I think, David, that's a real issue for us. Any of the traits we come up with, they have to have a large impact in order to warrant the cost of getting them through all this regulatory, system. It really, with editing, maybe we won't have to do that. But currently there's still international regulations to be concerned with.
Yeah, so that's a good point. What you're saying, somebody might say, in this particular part of in my part of the world, this is a big issue. Mm-hmm. And as a company, we may have to say, well, I understand that's a big issue, but we don't have hundreds of millions of dollars in a decade's time to solve that very specific small issue, right? Because in order to recoup our cost of investment and research, we really have to work on the big picture projects.
Yeah.
Is that fair?
Very true.
Very, very true. I think that's something when I was younger, I had no idea about. We looked at biotech and thought, wow, but the regulations part of it, that was something that came about later and just seemed to grow and get more complex, as it involved the whole world, basically.
And to be fair, I think it's changed, right? At one time I remember reading about, when original Roundup Ready soybeans came out in the mid-'90s, the list of countries you had to get approval in, and I want to say it was 14, 15 or something, and that's 2x and maybe 3x today for a product you want to release today, at least in broadacre crop. So I think it's just inherently become that much more complex as time went on. But well, that's exciting. So gene editing may have a prospects for creating additional products that would bring value to those growers. The other thing I guess I'll ask you, I think Bruce had kind of as an example talked about yield enhancement, right? So I'm sure there are certain processes that are easier than others, right? So would it be fair to say when people start talking about, we're going to try to map, the parts of the genome that relate to yield, in my mind, I picture that being an extremely complex thing because I'm sure there's a lot of interactions. Is that fair? Is that a correct assessment?
Yes, I think that's very fair assessment. I think the complexity of the genome and the interactions are just oftentimes beyond our comprehension or just very, catch you off guard because you think you did something, but oh no, something else influences it, and it doesn't work like you planned. So it's very complex to especially, a lot of these traits, even simpler traits that we've worked on, that you think are maybe single gene traits or, they turn out to be multigenic or bigger than you thought. They involve maybe 100 genes that you weren't aware of or something like that. So it can become complex. So you can imagine yield, how complex that is.
Yeah, I would think there's just a lot of interactions that take place there that would be difficult to come up with the whiz-bang thing that's going to create, inherently higher yield, right?
There's been some companies that have approached us and said, hey, we got this gene and it makes larger seed. It's going to increase yield. Well, larger seed isn't what increased yield is. You got to put it on an acre. They don't understand the concept of, it's weight per acre. And it doesn't, larger seed doesn't mean you're going to get more yield.
Right.
What if you get less seed or whatever?
Fewer seeds or whatever.
Yeah. So it's kind of interesting. Even the concept of yield a lot of times out there isn't the farmer or, isn't what we deal with, when it comes to yield.
And that's where the benefit of Stine comes in probably because you guys know that and you can take it from the biotech through the development, through the elite trial testing, and eventually, if it works, we put it in a bag and we can sell it to farmers. I think that's fantastic. So as we kind of wrapping up the conversation, I guess one question I have in my mind I was going to ask you is, Sekar, I guess I'll start with you. What is something that you wish that our farmer customers knew about what it is you do?
Yeah, just like what I mentioned before, this is so precise as science. In order to come up with a new trait, discovery of the trait, and then from whatever source, and then introducing it to a plant and making that trait to work in that plant and screening and testing and making sure the trait is to the level that you are expecting. This all takes a long time. And so from the lab to the field as a product, it may take a decade or more. And that's one thing that farmers need to understand. And it's a lot of work actually. One of the things farmers may be surprised is, you may have heard Bill say, over 90 or 95% of the projects that we work with don't pan out to anything. So we'll be lucky if 5% or in that ballpark, if 5% of our projects clicked to a certain extent, that'll be a big reward for us. And so it's a lot of even though it's a precise science, it's still a lot of trial and error, and so it takes a lot of effort and time.
Yeah. How about you, Bruce? Thoughts on that?
Yeah, we've had some farmers come through our lab, and I just like to tell them how the nature of what we do is so different than the nature of what they do. I'm from a farm originally, and, farmers work with large equipment and, big acres and that kind of thing. And what we do is on such a micro scale. We work with small things, nanoliters, microliters, micrograms, nanograms. And when we work with DNA itself, you're working with something you can't see, with the naked eye, and you always have to analyze it to know what you're working with, but you're actually working with something you can't see. So the nature of it is so different, that way we work with such small things, really.
Yeah.
Yeah.
Yeah, it's a whole different scale.
Yeah. And, I like to expand just a little bit on what Sekar says. What we do, we fail a lot, and that's the nature of research, right?
Right.
But I feel it's a really, it's a privilege to work in that area and to actually be able to fail, I guess. That's something, it's kind of interesting, that oftentimes I think, failure comes with, success, so,
Yep.
You fail a lot. So it's a bit of a privilege to actually have that opportunity to work, in that environment.
Well, and as you alluded to, over the years, I know I've brought a lot of farmers to visit you guys up in Ames to tour the lab. And, just like me, they don't always understand everything that's going on there, but it's fascinating, and it's and they come away with an understanding of the seriousness of the work and the level of work that's being done. And we appreciate that. And I think the other thing I was just going to say, Bruce, I always appreciate you, you guys understand the end use of the product. Bruce will call me, 2 or 3 times a year, and he'll say, "How are sales doing? What are the products? What are farmers buying?" And I want you to know that I think we appreciate that, because, I think you don't just say, well, okay, we've done our little part of the project and now it's gone. You care about where that ends up and how it's getting used. And I think that's a fantastic testament to the work that you guys have done there at the lab. And so, anyway, appreciate having you here. It's been a great conversation. I really, really appreciate what you do for Stine, and wish you much success in the future. And we'll want to have you on again and visit about new things. So thanks to you both.
Thank you,
Thank
David.
you, David.
That's our time for today. I'd like to thank our guests and listeners for joining us for another episode of the Stine Seedcast. We'll be back again soon with more expert interviews and insights about all things Stine. And to never miss an episode, subscribe to the Stine Seedcast wherever podcasts are found.
Subscribe to the Stine Seedcast wherever podcasts are found. To learn more about Stine and its elite corn and soybean genetics, visit stineseed.com. Stine has yield.